and a radiation oncology residency and research fellowship at Harvard, Dana-Farber Cancer Institute, and Broad Institute in 2019. He's a physician scientist and sees brain tumor patients in the Duke Radiation Oncology Clinic. His research lab has received more than $4 million in funding from the NIH philanthropic and foundational resources, including Alexis Lemonade Stand Foundation A Award. His work has identified frequent brain tumor mutations, defined brain tumor heterogeneity, and identified novel potential therapeutic approaches, leading to lead author publications in journals such as PNAS, Nature Chemical Biology, Journal of Clinical Investigation, Nature Genetics, and Nature Communications. The floor is yours, sir.
Thank you. Well, so glad to be invited to give a Terasaki talk, and I'll jump right into it. As mentioned, my research lab focuses on brain tumors, and we're interested in a few different topics that I'll touch on in this talk. On the left, we're interested in brain tumor omics using genomics, transcriptomics, and other high throughput analysis methods to better understand tumor heterogeneity and treatment response. At the top, we're very interested in genetic mouse models, designing new models and characterizing them to better identify new treatment approaches. We have an interest in targeting telomere biology that I'll touch on a bit in this talk, such as the TERT promoter mutations found in brain tumors. We're very interested in correlating outcomes for brain tumor patients with genetic and immune features to better understand determinants of treatment response and radiosensitivity. And we have a special interest on the bottom right in using cancer-derived enzyme mutations to guide the design of novel enzymes for a variety of purposes. In this talk here, I'll talk about what got me into oncology and science. The first part of the talk will be about the mutational landscape of glioma, my early work during my MD PhD studies. The second part is some of my postdoctoral studies that focused on multi-omics and mouse modeling of brain tumors. And then I'll showcase some of the recent work in my lab at Duke, leveraging mouse models, multi-omics, and genetic analyses of brain tumors to identify new treatment approaches. So I was exposed to oncology more than 20 years ago. My father was a 45-year-old electrical engineer who was diagnosed with multiple myeloma, which is a cancer of the bone marrow. And he underwent several different chemotherapies and systemic therapies, and in 2003 developed weakness and paralysis of his legs. And he was found on an MRI scan to have spinal cord compression caused by a tumor in his thoracic 5 vertebrae, shown here on a sagittal MRI scan. There's a tumor coming out of the bone and compressing the spinal cord. And so I got my first exposure to radiation oncology, which is the specialty I practice now. And the radiation oncologist offered my father a course of radiation therapy to the spine. 30 gray and 10 fractions of radiation energy beams were directed at the T-spine over about 2 weeks. And this melted away the tumor in the thoracic spine, and my father regained the ability to walk. He weaned off steroids, and the follow-up MRI scan looked pretty good without much evidence of tumor in that area. And so this got me motivated to go to medical school, to study oncology, and to get into this line of research that I'll talk about that's motivated me for the past 20 years or so. But I knew that myeloma was a little bit too close to home. And so during my medical school years, I became interested in brain tumors, in particularly primary brain tumors that arise in the brain itself, as opposed to secondary brain tumors or metastatic brain tumors that come from a primary site elsewhere in the body. These account for about 88,000 cases per year in the US. They're the most common solid tumors of children. They affect many young adults, young people in their 20s, 30s, and 40s, like my father. The most common malignant subtypes that I'll talk about here are the gliomas. And one of them is shown in the MRI image on the right. Standard of care for many of these involves resection by neurosurgery, radiation therapy, and chemotherapy. But nevertheless, these cause more than 16,000 deaths per year. And so there's a desperate need for improved classification and treatment for brain tumors. And this inspired me to do my PhD in the lab of Hai Yan at Duke, where at the time we were uncovering the mutational landscape of brain tumors. We discovered key mutations in gliomas, the metabolic functions of some of the mutations, for instance, in the enzyme IDH1 that I'll mention. We studied the mechanisms of telomere maintenance in brain tumors, and we learned about the brain tumor mutational spectrum in childhood brain tumors, which is different from that of adult brain tumors. When I was joining the lab around 2008, 2009, Hai and his colleagues were just discovering these frequent isocitrate dehydrogenase mutations in gliomas. And the mutational spectrum is shown here. On the left, these frequent mutations in arginine 172 of IDH2, which is the mitochondrial isocitrase enzyme, were found in about 5% of certain glioma subtypes, shown in the red bars on the right. And about 90% of some of the grade 2 and grade 3 glioma subtypes had IDH1 mutations. This is the cytosolic isocitrate dehydrogenase. Those mutations occurred in arginine 132. And what you could see is these IDH1 and IDH2 mutations helped classify these brain tumors. Basically, they are pathognomonic for the grade 2 astrocytomas and oligodendrogliomas, and they exclude some of these other subtypes that very rarely have IDH mutations. And so this was an interesting finding that emerged through exome-wide sequencing and genetic studies, but at the time, it was unknown what the function of these IDH mutations was. And so work at Duke and University of Pennsylvania and other institutions uncovered that these IDH mutations confer a metabolic gain of function to the isocitrate dehydrogenase enzymes. Here on the left is a review I helped write in Cancer Cell, illustrating the phenomenon. Basically, the wild type enzymes take isocitrate, they decarboxylate it and oxidize this alcohol to a ketone group, whereas the mutants reduce the ketone group shown on alpha-ketoglutarate at the bottom to an alcohol and create a metabolite called 2-hydroxyglutarate. And we did a global metabolite profiling study using mass spectrometry-based techniques to profile about 300 metabolites in human glioma cells and found on the right on this principal component plot that IDH enzyme expression of these cancer-derived mutants results in a very different metabolic profile in the cells that's characterized by 2-hydroxyglutarate levels being quite high. And having learned this biochemical insight about the IDH mutations, there was a great need to develop model systems to study these mutations. And so folks in the Yan lab and elsewhere were busy developing mouse models that took years to develop. And during my PhD, I developed a fly, Drosophila melanogaster model of the IDH mutations, in which we used this UAS-Gal4 system to provide tissue-specific expression of fly isocitrate dehydrogenase mutants that had the cancer-derived mutations in them. So this Drosophila IDH on the bottom has arginine 195 or arginine 163 mutated. These are equivalent to the human mutations found in cancer. We showed that these cause phenotypes in the flies, such as melanotic tumors in the larvae, neurologic deficits, proliferation of brain progenitor cells, and high levels of R2 hydroxyglutarate from the enzyme activity. So it provided an early way to study the phenotypes associated with IDH mutations and dissect the genetic pathways. But as I mentioned, mouse modeling and other mammalian systems were needed to better model the human condition. And so much has happened since that work in 2008 to 2010, and IDH mutations have now revolutionized glioma treatment. There are inhibitors that have been developed by Agios Pharmaceuticals and others that inhibit the mutant IDH enzymes, and they've now received FDA approval after exciting results published in the New England Journal of Medicine. Here's a group of patients with grade 2 gliomas with IDH mutations, and the patients who received an inhibitor of the IDH mutation survived much longer before they required another intervention for these tumors. Another area of interest shifting from the IDH mutation was how do glioblastomas and other brain tumors elongate their telomeres? It had long been known that brain tumors inevitably find some way to elongate the telomere ends at the end of their chromosomes, which is thought to help cells achieve immortality and avoid the Hayflick limit of a finite amount of replications. But the mechanism by which this happened long remained unknown. And during the time I was a PhD student, 2 papers came out in Science that identified highly recurrent promoter mutations outside the telomerase reverse transcriptase gene in melanoma that seemed to activate the promoter and turn on transcription of TERT, the telomerase reverse transcriptase. And so immediately in February 2013, when these papers came out, I thought perhaps a similar mechanism could be at play in gliomas. Here's a zoom in of the mutations seen in melanoma. They occur at these 2 different sites that are 228 base pairs or 250 base pairs upstream of the transcription start site at the TERT locus. And they create these novel transcription factor binding sites for these 1110 domain transcription factors, which then are thought to bind these sites and turn on transcription of TERT. And what I found working with another graduate student named Patrick Killalay was that the TERT mutations were in fact very common in glioblastoma and other gliomas. This chart shows the frequency. Greater than 80% of glioblastomas and oligodendrogliomas and other brain tumor subtypes had these important TERT promoter mutations in the tumors. And so together with our studies of the IDH mutations, the TERT mutations, and other alterations that we helped uncover, such as those in the ATRX Swiss SNF complex protein that's involved in an alternative lengthening of telomeres phenotype, 1p19q chromosomal co-deletion, p53, tumor suppressor mutations, we were able to put together a molecular classification for gliomas. And what you can see here is grade 4 gliomas can be classified as a classic glioblastoma by the presence of a TERT mutation and absence of an IDH mutation or these other features. Whereas if you have an IDH mutation and a TERT promoter mutation or an ATRX mutation, you may fall into an astrocytoma or oligodendroglioma classification as shown here. And so I use these classifications every day in my clinic to guide dose selection for the patient's radiation therapy treatments, clinical trial enrollment, and counseling for prognosis of my patients. So to summarize part 1, in my PhD studies, we uncovered key mutations that have redefined glioma classification. These included IDH1 mutations that reprogram the metabolome and ATRX and TERT mutations that define telomere maintenance mechanisms in brain tumors. Which is all very nice, but at the time hadn't done anything whatsoever therapeutically for the patient. So I was still very interested in seeing how we could take these genetic discoveries and improve patient care. And so I went on to my postdoc at the Harvard Radiation Oncology Program, the Broad Institute, and the Dana-Farber Cancer Institute, and further postdoc training at Duke with some really good mentors. This is Pratiti Bandyopadhyay at the top, who is an associate professor of pediatric oncology at the Dana-Farber, and David Kirsch at the bottom, was then a professor of radiation oncology at Duke and since has become the chair of radiation therapy at Princess Margaret Hospital. And these mentors and others helped me learn a lot of functional techniques to help answer some of these questions. How do mutations drive tumorigenesis? How do aggressive brain tumors differ from benign tumors? How do these brain tumors resist therapy? One concept that interested me throughout this period of my training and to this day is the idea that pediatric brain tumors seem quite different from adult brain tumors. Here in 2009, I published with Hai Yan, my PhD advisor, a short report in the New England Journal where we found that children with intermediate grade gliomas did not have IDH mutations in the tumors, which is quite puzzling. Tumors that look exactly the same but come from adult or teenage patients did have IDH mutations in them. And this, along with decades of further research, have uncovered that pediatric tumors are wired differently. They have different mutations in general from the adult tumors. And so there are key differences that perhaps could be exploited or at least used to better classify these tumors. Here's another diagram that I put together where on the left are two common childhood glioma subtypes that are characterized by mutations in histone genes. For instance, at the very left are the diffuse midline gliomas that frequently have histone 3 gene mutations causing lysine 27 to methionine mutations, or other mutations in histone genes, such as glycine 34 to arginine or valine mutations. And what you can see here is that these childhood brain tumors have different anatomic locations in general. For instance, on the left, the diffuse midline gliomas tend to be in the midline, and they have a really poor prognosis. And all of these things differ from adult brain tumors that have a different constellation of genetic features shown here. They occur in different regions of the brain, and they may have different prognoses. And so as I became interested in these pediatric gliomas, spurred by Dr. Bandyopadhyay, who's a pediatric neuro-oncologist, I became interested in the entity known as the diffuse midline glioma. These are deadly tumors. They occur in children and young adults. They occur in the brainstem or thalamus or other eloquent areas where gross total resection is impossible. And chemotherapy seems to be ineffective for most of these tumors. That leaves radiation therapy, my specialty, as the standard of care, but this is essentially palliative. The tumors inevitably recur. And so there was interest in biopsying or obtaining samples at autopsy from these deadly glioma subtypes. And one of the studies I worked on during my PhD and postdoctoral years with Dr. Yan and others was an exome-wide sequencing study of diffuse midline gliomas. Here's some of the data from one of these studies. Each column in this oncoprint is a different brainstem or thalamus glioma, what are essentially now known as the diffuse midline gliomas. And we uncovered some novel findings here. For instance, we saw that about 20% of these tumors have a mutation in a gene called PPM1D, which was not known before our exome-wide sequencing study. And the PPM1D mutations occur in a protein or a gene called protein phosphatase 1D, manganese and magnesium dependent. And these encode a phosphatase. And one of the most striking things we noticed at first sequencing of these tumors was that all of the mutations in the gliomas in PPM1D occurred at affected the C terminus of the protein. And they were all truncating or frame shifting or nonsense mutations that would be predicted to cleave off the C terminal end of the protein. And what's been worked out is there's a putative degron in the C terminus of PPM1D in the tail. When it's removed, the protein becomes hyperstabilized, and the phosphatase domain, which is really the active enzymatic area of the PPM1D protein, remains intact and is inappropriately present and active in cells in a gain of function fashion. And so as we discovered these mutations, we looked up the function of PPM1D, which in normal cells helps to return the cell to homeostasis after DNA damage. For instance, when a cell receives ionizing radiation or other genotoxic insults, numerous cell cycle checkpoint genes are activated. For instance, DNA double-strand breaks cause phosphorylation of histone H2AX, known as gamma H2AX. They lead to autophosphorylation of the ATM kinase. They lead to phosphorylation of P53, CHEK1 and CHEK2, and these lead to cell cycle arrest at the G1 and G2 cell cycle phases. PPM1D is expressed downstream of P53, the notorious tumor suppressor and transcription factor, and comes in and dephosphorylates all of these cell cycle checkpoint proteins and helps the cell to return back to the cell cycle, presumably after it's finished repairing its DNA. And so it stood to reason that these PPM1D mutations in cancer were somehow inappropriately leading brain cells to divide and proliferate, perhaps in the presence of genotoxic stress, in a way that ultimately led to tumor formation. And during my postdoc, I worked with Prasida Kadka, a talented Harvard graduate student, now medical student at UCLA, to do a number of studies on PPM1D, where we found that these PPM1D mutations did confer a growth advantage to glioma cells. And we also saw that if we inhibit PPM1D with an inhibitor that's been developed by GlaxoSmithKline, we could slow the growth of PPM1D mutant diffuse midline glioma cell lines, as shown here. And if we added ionizing radiation to the PPM1D inhibitors, we could further slow the growth of these organoid cell lines. So this revealed an intriguing mechanism that is likely involved in tumorigenesis of the diffuse midline gliomas and likely may be associated with actionable therapeutic vulnerabilities. But a number of questions remained. One question is, how do these diffuse midline gliomas that are so deadly, median survival of less than 2 years after diagnosis, differ from other pediatric low-grade gliomas that are associated with a much better prognosis and more indolent disease course? For instance, the pilocytic astrocytoma is characterized by fusions that involve the BRAF oncogene, often with the KIAA 1549 gene. And these tumors are often curable by surgical removal alone. They're low grade, they grow a couple of millimeters a year. It's a very different clinical story from the diffuse midline gliomas, which, as I mentioned on previous slides, have a very different genetic composition. And so during my postdoc, I set out to do some of the first single-cell RNA sequencing experiments to define the heterogeneity within these different brain tumor subtypes. In particular, I applied single-cell RNA-seq approaches, which were really brand new at the time, 2016, 2017, 2018, to 6 pilocytic astrocytomas that had been obtained in the operating room of Boston Children's Hospital. And we uncovered a transcriptional developmental hierarchy in this brain tumor type. Here I've displayed single cells from these 6 different pilocytic astrocytomas, and each dot reflects the transcriptional state of the cells based on their RNA-seq profiles, and the orientation of the cell represents whether the transcriptome is more similar to an oligodendrocyte progenitor cell, if it's closer to the top of the axis, more like an astrocyte, which is a normal supporting cell of the brain towards the left, or more like an oligodendrocyte, which is another axon supporting support cell in the brain. And we could see that there's a whole spectrum of different transcriptional states within these tumors, reminiscent of other glioma cell types. And we could see that most of the cells that are cycling and dividing are more progenitor-like, but also that many of the cells that are progenitor-like are expressing signals of cellular senescence and appear to have exited the cell cycle, perhaps alluding to why these brain tumors are more indolent, slower growing, and in many cases curable, when some of the others simply are not. And so this schematic illustrates some of the differences we notice between different glioma subtypes. On the right is the pilocytic astrocytoma that I displayed on the previous slide. The tumor cells transcriptionally look a lot more like mature glia and less like stem cells, though some of them seem progenitor-like. They have relatively few cycling cells, and some of them seem, if anything, senescent. And that's quite different from astrocytomas of adults or the diffuse midline gliomas that I showed a few slides ago, where the cells seem a lot more primitive or neural stem cell or radial glia-like. There's a lot more cycling cells and less cells exhibiting a senescence pathway. So it clearly links developmental hierarchies and developmental processes to differences in these different brain tumor subtypes. But I knew as I carried on these genetic and transcriptomic studies that there's a critical need for models to study tumorigenesis and treatment in vivo. Questions arose in my mind as to how do these transcriptional developmental hierarchies develop during tumorigenesis? How do mutations in the tumors affect these hierarchies? How do they affect the vasculature? How do they affect the immune microenvironment in the tumor? And how do different mutations affect the treatment response? And to answer many of those questions, we need models that are primary, meaning they spontaneously arise in the mouse or model organism so that you can study the process of tumorigenesis itself in an autophonous manner. To study the tumor immune microenvironment, or perhaps even the tumor vasculature or a faithful treatment response, you need models that are immune intact. And there's a great need for models that are amenable to genetic manipulation. And so this motivated me to move from Harvard up to Duke and to do additional postdoctoral training with David Kirsch, who I mentioned earlier and is shown here, and also with Catherine DeLand, a talented senior postdoc at the time, and Oren Becker, a pioneer in the field of mouse modeling of pediatric brain tumors. And I worked with them on a study shown here where we used genetic engineering approaches to engineer mouse models to dissect tumorigenesis and treatment response. In this particular effort, we introduced alterations in the p53 tumor suppressor to neural stem cells in mice using the Nestin TVA and ARCAS TVA retrovirus-based mouse modeling system. And then we also deleted key radioresistance-related genes such as the ATM kinase in the vascular cells of the tumors. And we were able to answer questions as to whether radiation responses in these mouse tumors were more the effect of radiation killing tumor cells or radiation killing endothelial or vascular cells. And the conclusion from this work was that it's the tumor cells. It's not that radiation is killing vasculature as the primary therapeutic drive for this treatment. So to summarize part two, PPM1D mutations deregulate the DNA damage response and drive diffuse midline glioma formation. Single-cell approaches have revealed tumor cellular hierarchies that resemble developmental processes. And genetic mouse models can perturb specific tumor compartments during tumorigenesis and treatment. And so taking all of this genetic observation, mouse modeling, single cell and other omics technologies into my tool belt, I established the Reitman lab about 5 years ago. And our goal is to use these tools to improve the efficacy of radiation therapy, to find new ways to target brain tumor mutations, and to better understand tumor heterogeneity to improve treatment approaches and overcome treatment resistance. We have a special focus on genetically engineered mouse models, or GEMs. And it's for many of the reasons that I mentioned a few slides ago. These are immune intact models that allow you to study tumorigenesis. Other model types that I've shown above, such as patient-derived xenografts, syngeneic mouse models, and so forth, are critically important. And we do use them in our lab, and they are needed to complement GEM studies, or in some cases, they are the best model for specific questions. Here I've reiterated some of our motivations to be interested in the primary genetically engineered mouse models to study brain tumors. We're very interested in how specific mutations drive tumorigenesis and progression. How does the tumor developmental hierarchy arise that we observed in human brain tumors using single-cell approaches? We're interested in understanding how treatment affects the developmental hierarchies we've observed and what populations seed recurrences. And this whole lower part of the slide involves examining the tumor immune microenvironment, which I would argue is critical to study in primary models where the tumor co-evolves with the vasculature and the immune system and so forth. And this is particularly critical because immunotherapies have gained great traction in many cancer types. There's hope that they could be effective in brain tumors. So far, they have mostly been unsuccessful, but careful study of appropriate models are needed to understand how to unlock efficacy for immune therapies. A key technology in our lab is our image-guided focal brain irradiation. Here is a protocol paper spearheaded by Lauren Weidenhammer, who was a research technician in the lab, now a graduate student in our department. These are images from our SARP, Small Animal Radiation Research Platform, where we've developed workflows to immobilize the mice, shown in A, under anesthesia, perform image guidance, shown in B, to localize our radiation beams, and then we can deliver conformal radiation beams. Here are isodose lines shown on a sagittal cone beam CT image of a mouse, demonstrating that we can deliver the radiation very tightly to the brain area shown in red and avoid the salivary glands, mouth, nose, esophagus, and so forth, which is critical for getting safe dose in and modeling conformal radiation therapy in these mice. Panel D shows a dose volume histogram, which is similar to what we use in the clinic, showing that the parotid and submandibular glands receive relatively little dose compared to the target in this setup. Vanessa Valentine is a graduate student in my lab who's been working with others to develop a novel mouse model of the PPM1D mutations that I studied during my postdoc at Harvard. Here we've developed a unique mouse allele and engineered it into the mouse PPM1D gene locus on chromosome 11, where we introduce LOX-P sites around the exon 6 of PPM1D. This allows Cre recombinase to go in a conditional manner and delete the wild type C-terminal encoding exon 6 of the PPM1D gene. We then have a splice acceptor that splices on a mutated exon 6 containing a DMG, diffuse midline glioma-derived nonsense mutation with an epitope tag. And Vanessa has expressed this in our mouse model and shown that compared to litter-mate controls, mice that have this PPM1D allele and that are exposed to the Cre recombinase through the RCAS-TVA retrovirus delivery system have a faster development of brain tumors than controls. And she presented some of this work at the SNO annual meeting, Society for Neuro-Oncology, last year, and we're characterizing these mice and preparing a manuscript on the results. We've also been combining this retrovirally based RCAS-TVA system with CRISPR-Cas9 approaches. The RCAS-TVA system that I've alluded to throughout the talk is an avian or bird retrovirus system, which lets us use bird retroviruses that we inject into the mouse brainstem to deliver different genetic payloads. But it hadn't been used to deliver CRISPR-Cas9 guide RNAs to model diffuse midline gliomas before. And Sophie Wu, a research technician and recent undergraduate graduate from Duke, from my lab, developed a mouse model that's able to do this. It expresses Cas9 in Nestin-positive neural stem cells. She's able to inject RCAS retroviruses that contain guide RNA payloads into the mouse brainstem. And she's able to use next-generation sequencing to observe genetic disruption of different genes that were targeted by the guides, such as ATM, tumor suppressors such as P10 or P53 or CDKN2A. She was able to use barcodes that we put on the retroviral vectors to count how many of each guide construct was taken up by the different tumors. And on the right, we could see there's a strong correlation between what area of the genome was disrupted by CRISPR and the representation of the different barcodes. And so this is a nice system. We just published it a few months ago, and we hope this will streamline future experiments and enable experiments where we do in vivo screens, where we pool different perturbations together and ask which perturbagens better drive tumorigenesis, which cause resistance or susceptibility to different therapies and so forth. Speaking of therapy, one of our biggest therapeutic interests is targeting the ataxia-telangiectasia mutated kinase to enhance the efficacy of radiation therapy. A number of clinical trials have been initiated with inhibitors of the ATM kinase. This is an apical kinase that detects double-strand DNA breaks and orchestrates, by phosphorylating many targets, shown here, the cellular homologous recombination-based response to these insults. And compounds such as AZD1390, developed by AstraZeneca, have entered clinical trials for adult brain tumor patients in combination with radiation. And we've long wondered if these could be effective in the diffuse midline glioma population, the children and young adults with this deadly brain tumor subtype. And so our group has used one of our RCAS-TVA-based mouse models in which we've deleted the ATM gene within the tumors in the blue group here and compared this to littermate controls in which there was an intact allele of ATM in the tumors. And we saw significant radiosensitization when the mice were subjected to whole brain, focal brain irradiation. This work was led by Avni Mangoli, one of my pediatric neuro-oncology colleagues, and others, and builds up support for deploying ATM inhibitors in these pediatric glioma patients. We've been applying multi-omic techniques that keep evolving to our mouse models. Here's a diagram that shows several different technologies that are now available through 10x Genomics. These include at the top single-cell RNA sequencing, which gives you single-cell resolution and the full transcriptome, but it lacks certain information about tumors, in particular, spatially resolved information. It doesn't tell you where the cells are or what cells are next to each other and so forth. And so one exciting technology that can help with this is called Visium, which is spot-based RNA-seq in a spatially resolved manner, which can give you spatially resolved data and give you the full RNA-seq-based transcriptome. However, Visium doesn't do this at single-cell resolution. Another complementary technology shown at the bottom is the Xenium in situ sequencing platform from 10x Genomics, which does give you single-cell resolution. You can profile individual cells with this, and does this at a spatially resolved level. And it even preserves the sample for future immunofluorescence and so forth. One drawback is it doesn't give you full transcriptome of the cells. It only can sequence a panel of several hundred, soon to be several thousand transcripts per cell. And so we've been applying this technology to some of our brain tumor models. Here, for example, we applied it to our mouse glioma model. You can see this tumor circled here. And you can then ask in a more focused way, what genes are differentially expressed, not only in the bulk of the tumor, but specifically in neoplastic tumor cells. This can avoid having findings that are masked by tumor-resident normal cells, such as vasculature or immune cells. And so here we saw that stem-like markers and known radiation resistance factors such as CDKN1A, tumor suppressor, are top differentially expressed genes after radiation therapy in this mouse model. We've applied some of these multi-omic techniques to a number of different brain tumor types. For instance, Josh Riegel here, postdoc in my lab, who is now faculty at Roswell Park Cancer Center, applied some of these spatially resolved transcriptomic techniques to an interesting tumor called ganglioglioma. These tumors have long been known to have both neuron-like and glia-like components, and some have speculated for decades that there may be these protoplastic astrocyte-like cells within the tumor that could be serving as stem cells that give rise to both the neuron and glia-like components of the tumors. And this work is now published 2 years ago, but what Josh worked out is that if you look at cytotrace pseudotime and signaling entropy rates as approaches to identify stem-like cells in these tumors, you can find a subpopulation that seems to track with these protoplasmic astrocytes, and that express a neuroectoderm-like neural stem cell gene program, indicating that perhaps this is a cell of origin for this unique and somewhat mysterious brain tumor subtype. This may inform classification of these tumors or future therapeutic approaches. Another longstanding interest that I mentioned earlier is my interest in the TERT promoter mutations that I helped discover years ago as a PhD student. I was always unsatisfied because I wanted to find ways to leverage these mutations to come up with new treatments for patients. One idea I've long held is that perhaps if you inhibit something upstream of the novel, the promoter mutation that recruits aberrant transcription factors to the promoter site, that you could turn off the oncogenic activity of the TERT promoter mutation. And so I worked with Kevin too, a talented Amgen scholar in my lab, now a medical student, to perform pooled genetic screens where we compared TERT promoter mutant cell lines to TERT promoter wild type cell lines. And we saw a number of differences both at the transcriptomic level and in their genetic vulnerabilities as assessed by CRISPR screening approaches at the Broad Institute DepMap project. One of the important findings that I'm showing here that's in one of our recent publications is a transcription factor-based analysis of differentially expressed genes showed that ETS transcription factors underlie many of the differentially expressed genes seen in the TERT promoter mutant cell lines compared to controls. Here's some examples of the ETS transcription factors. And what this brings up, and what was sort of the conclusion of our study, is that these TERT promoter mutations likely subvert an overlapping and complex milieu of different transcription factors to carry out their oncogenic functions. And it may be difficult to simply target one molecule or one transcription factor to fully kill glioblastoma cells. Wrapping up, I'm working on a number of other exciting projects and collaborations, including analysis of circulating biomarkers of brain tumors, analyses of ultra-high dose rate flash radiation therapy, where when radiation is delivered very quickly, it seems to have unique properties that could stimulate the immune system or protect normal tissues from radiation damage. And we're interested in the insights gained from cancer mutations in enzymes, such as PPM1D or IDH1, to guide the design of new enzymes that could be helpful for chiral drug synthesis or bio-based production applications. So to summarize, my lab is using multi-omic techniques to discover unique features of brain tumors such as ganglioglioma. We're defining genetic subtypes of diffuse midline gliomas, and in particular, whether these might be susceptible to ATM inhibitors. We're doing work mapping outcomes of patients to the presence of different genetic variants, and we're developing different new primary models of brain tumors to better understand tumorigenesis and treatment approaches. And so it's been so exciting. You know, my father, I mentioned in 2003, really got me excited by cancer research and clinical practice. And I'm honored to have been around for discovery of IDH mutations, TERT mutations, the reclassification of glioma, and now new treatment approaches that are entering the clinic, such as IDH inhibitors and others. And I hope to continue to do more. And with that, I'll thank many in my lab and collaborators, funding sources, the patients and members of my lab who have made all of this work possible. And thank you all for inviting me to give this talk and to reconnect with old friends and colleagues.
Thank you, Professor Reitman. I appreciate the detailed seminar. As we wait for a couple questions, these mutations from a therapy perspective, are they random or is there any type of order within them?
Yeah, I think the mutations, I guess there's two pieces. One is how are the mutations caused, and two is how are they selected? And I suspect the cause is often somewhat random. In the brain, there usually aren't many carcinogens or mutagens. Presumably, a brain cell is just picking up a random mutation. And I think the thing that is not random is the selection that then occurs, whether it's a TERT promoter mutation, IDH mutation, PPM1D, whatever. For whatever reason, these are highly selected to probably expand some group of stem cells in the brain that could then grow into a tumor. You know, that's my thought.
Thank you. What are your thoughts on treating IDH mutant high-grade glioma with IDH inhibitors? Are there any opportunities to enhance their effectiveness through combination therapies?
Yeah, this is a big question. It comes up quite often at Tumor Board and in my clinic, because as the questioner alluded to, the clinical trials are in low-grade gliomas, and many have tried to push the envelope to high grades. anecdotally, it doesn't seem to have great outcomes. If the patient has a high-grade tumor, they don't tend to respond, at least not for very long. I do think there are opportunities to combine the IDH inhibitor, for instance, with radiation or with chemotherapy, the things that we know work. And I think that needs to be carefully done with clinical trials. And I think the final piece I'll add is I'm very interested, and I think it's very important to think about sequencing. We don't know, with all these patients receiving IDH inhibitors, if other treatments that are quite effective will still work afterwards. There are big clinical trials of radiation and chemo for low-grade gliomas that show huge overall survival benefits. IDH inhibitor has never showed an overall survival benefit, just a radiographic and time to next treatment benefits. And I think a lot of caution is needed and a lot of thought about sequencing, making sure all the therapies still work in a certain order is highly warranted.
Thank you. I read a paper, it said large animals, they have this Peto's paradox, they tend to evade cancer. Do you think we still can learn things from nature about treatment or even prevention?
Yeah, the short answer is yes. I think large animals. I think of elephants, whales. I mean, even the naked mole rat. They have mechanisms to avoid cancer, and I think much can be learned from that. Clues emerge. Do those animals have extra copies of P53? Do they have other mechanisms? I think it informs our understanding of tumorigenesis, and I think a lot can be learned from it. A lot of my work, especially the enzyme redesign work, has taken inspiration from evolutionary processes, whether it's cancer or evolution of species. I think there's a lot there that can help us understand things, basically.
Thank you. Has there been any recent advances in developing novel therapeutics by targeting TERT promoter mutations? Can you comment the roadmap for treating diffuse midline gliomas?
Yeah, recent advances in treating TERT. So in addition to the work that I showed, a few other labs are making exciting progress. Dr. Costello at UCSF has done some important work that shows that targeting the GABP transcription factors could be a promising avenue. The work I showed here raises a little bit of caution that there might be some redundancy there. Careful thought is needed. Work by Matthew Meyerson and his group and others have shown that perhaps there's some considerations that are needed in terms of tumor burden. And what I mean by that is perhaps if a tumor has been removed and resected and debulked down to a small number of cells, perhaps that is the time to target the TERT mutations and prevent it from regrowing. Presumably, it would need many cell divisions to regrow, and maybe that's a time that you can have a window of opportunity to target TERT. I think much needs to be done to find the right targets and combinations and timing for the TERT mutations. Another area of interest I'll add about TERT is I'm involved in a collaboration with Dr. Jerry Shea at UT Southwestern and others that's using a metabolite called 6-thioguanosine that may specifically target TERT promoter mutant cells in a metabolic fashion. So I think there's a lot of approaches. I think it's important to sort it out, get them into clinical trials. Diffuse midline glioma, there are advances. A number of consortia are designing very creative clinical trials. The CONNECT consortium comes to mind and others. I like approaches that stratify patients based on their genetic subtype to different treatment approaches and so forth. I do think ATM inhibitors are a prime candidate for these tumors, among other DNA damage response targeted agents, which are already in trials for other indications.
Thank you. The other questions came in. Truly appreciate for the fantastic talk, considering that an intact immune system is required for certain in vivo studies. How do you view the limitations of mouse models in accurately representing human genetics, given the inherent differences between marine and human immune systems?
Yeah, these are the challenges. A primary mouse model has the disadvantage of not being human and having a small anatomy and many other things. And even though I've made a strong pitch for the genetic mouse models and the primary mouse models, and that is what my lab puts a lot of its energy into, at the end of the day, I think you need lots of models and complementary models. And it's important to validate observations from the mouse model with a human model, perhaps a xenograft with a humanized immune system or things like this, which can be resource intensive, but important.
Thank you. To your knowledge, has depth sequencing or any other large-scale screening approach been applied to identify transcription factors that interact with the TERT promoter, enabling the comprehensive analysis beyond single target methods like chip sequence?
I am aware of numerous analyses that have looked at in vitro systems of what transcription factors interact with the TERT promoter, or in silico approaches like ours that link different transcription factors with the TERT promoters. To my knowledge, I'm not aware of human tissue-based studies of specific transcription factors. I'd have to go back and look.
Thank you. Following up on your single-cell transcriptomic study that revealed tumor cellular hierarchies, do we know what specific biological pathways or molecules are responsible for making certain cell subpopulations cycling or senescent?
Yeah, in the pilocytic astrocytoma, where I did the most work on the transcriptional developmental hierarchies, it was definitely the MAP kinase pathway. The oncogene in those tumors is an activated fusion form of BRAF, which naturally activates that pathway. Presumably, whatever the cell of origin is, probably some form of oligodendrocyte progenitor cell, is particularly susceptible to MAP kinase activation to cause it to grow. But I think probably that cell of origin or that lineage also has a strong senescence response. Once the MAP kinase pathway has been active for a while, or it's divided so many times, I think those cells are more primed to senesce perhaps than other cell of origins or configurations of genetic alterations.
Thank you. This person says, I really appreciate your work on H3K27M mutant gliomas. I was curious, have you looked into whether the impaired differentiation and gene expression changes are mainly due to the loss of H3K27 methylation, or could they also result from other effects of the K27M amino acid substitution itself? Have you or others done any rescue experiments to restore H3K27ME3 levels to see if that can reverse the phenotype?
Yeah, it's certainly been shown that the K27M causes dramatic changes in histone lysine 27 trimethylation, as mentioned. It decreases them through interactions with the polycomb repressive complex. We have not done those rescue experiments ourselves, but what we did do, and I encourage you to look at our recent paper in JCI, is we did whole genome bisulfite sequencing of tumors with and without the K27M mutation. And so this is genome-wide methylation analysis of the DNA, not the K27 methylation. And we did notice differences at the DNA methylation level that tracked with developmental pathways in the hindbrain. For instance, genes related to HOXD13 transcription factors that pattern the hindbrain were differentially methylated in our tumors. And so what I think that shows is it's not all lysine 27 methylation, which is thought to be the main driver by which K27 mutants exert their phenotypes. It likely also is conveyed by DNA methylation and probably a number of other epigenetic and other effects of the K27M mutants. So I think it is multi-pronged. We haven't done that rescue experiment ourselves, but that's very important.
That's all the questions I have. I appreciate your time, Professor.
Yeah, thanks. Very nice talking with everyone. Looking forward to chatting with some people next week. And thanks again for the invitation.
Thank you.
Thank you. Thank you, Zach.